The overall goal of this procedure is to produce biomimetic materials consisting of pure recombinant adhesins coupled to polystyrene beads, which can then be used to dissect the biochemical and functional interactions between individual bacterial adhesins and host cell receptors.
This method can help answer key questions in the field of host pathogen interactions, such as measuring the effects on host cell signalling and inhibition of pathogen-mediated cytotoxicity. The main advantage to this technique is that purified adhesins are directionally and covalently coupled to polymer particles that are similar in size to bacteria and thus mimic the bacterial surface display. Demonstrating the procedure with me will be Fitua Al-Saedi, a PhD student from our laboratory.
Thiol-amine directional coupling will be demonstrated here. This protocol is suitable for coupling of cysteine-containing proteins to amine functionalized polymer beads using sulfosuccinimidal 4-p-malimidophenol butyrate or sulfo-SMPB as a cross-linking agent. Prepare all necessary reagents just prior to use, as described in the protocol text. To begin the procedure for bead activation, mix the bead suspension by gently inverting and then transfer the required amount of bead suspension into a sterile 1.5 milliliter tube containing 1 milliliter of sterile PBS, pH 7.0. Gently pipette up and down to wash the beads and pellet by centrifugation in a microcentrifuge. Carefully remove the supernatant with a pipette and discard.